So can you hear me? If you can hear me, please type on your WhatsApp that you can hear me. Evans. Evans, please, if you can hear me, type. Let me know you can hear me. Hello, Evans. Can you hear me? Hello, Evans. Can you hear me? Evans, if you can hear me, please kindly type yes on your WhatsApp. Evans, if you can hear me, can you type yes? Okay. You're welcome. Lecture. With the 5th and the February 12 to 1. Would that you are set up already? Yeah, is she? Okay, wait for me. Okay, good morning, everybody. Let me see. So good morning, everyone.
Good morning.
Good morning, everyone.
Good morning.
Thank you very much. So I'm glad you can hear me so we can start the lecture at about now. All right, thank you very much. So thank you for coming, and I'm so grateful for putting your phones on silent today. Everything seems fine, so want to go ahead to microorganisms in the environment. And then we will also look at microorganisms in the industry, because we are supposed to have lecture for me for 3 weeks. 1 week is gone. Today is Friday, so we only have about 2 weeks, and I want us to end this lecture before 18th so that we can have time. So so we're going to look at the aquatic environment, you know, the environment made up of air, water, and soil. So we'll look at microorganisms in the aquatic environment first. Aquatic environments, of course, have various surfaces. We've started. Aquatic environments have varied surface areas and volumes, and they are found in diverse locations. You could find aquatic environments if the water in our body is an aquatic environment to some organisms. If you have organism in a bottle of water or drink or beverage, for example, that organism is found in an aquatic environment because that drink have the nutrients and the temperature or environmental factors that support the growth of that organism. So we also have the usual aquatic places like the river, the lakes, the lagoons, the oceans, the marines. Those are the vivid aquatic environments. And you know, we have various factors that is associated with different aquatic environments, ranging from acidic, alkaline, and different kind of, you know, some might be hot springs, some might be cold. You have heard about the Kogusi waterfall, where you have the mixture of hot and cold water can be associated with different environmental environments. The temperature within which organisms found in an aquatic environment can range from -5 to -15 degrees centigrade. So the lower range. And sometimes in very hot spring or hot geothermal environments, you can have as high as 121 degrees centigrade. We also have other factors like oxygen, nutrients, waste products, which can affect or dominate an aquatic environment. For example, in the lake or ocean, organic matter forms to great depth, where decomposition can take.
Where's the web?
Don't let anybody. Okay, understand you. Everybody they come from somewhere. You have to so if you work for this kind of thing, also give you how they fight around.
Evans, can you please control your class? Why? So you also have gases and soluble waste products that can affect microorganisms deep down the marine zone. You know, when we throw litters, physics, waste products, leftover food into the lagoon or into the ocean or rivers, after a while they get settled down deep into the sediments. And so you can have different organisms occupying the upper part of an aquatic environment. You can have different organisms occupying the lower part of the aquatic environment. For example, the oxygen diffusion in aquatic environment is limited. So only places where you have oxygen diffusion, that is where you can have aerobic organism. So you tend to have aerobic organism towards the surface of an aquatic environment, whereas you tend to have the anaerobic organism as you go down the ocean. And the ability of oxygen to diffuse through the water, through the aquatic environment, depends on so many factors. So it depends on the movements of the water, of the air. The ability of the air to mix and circulate will determine the kind of microorganism that you can find there. So oxygen diffusion in aquatic environment is limited based on the temperature and pressure. So solubility of oxygen depends on temperature and pressure. So when the temperature is high and the pressure is low, oxygen tends to diffuse faster in aquatic environments. Aquatic environments, like we said, excuse me. Okay, so like this diagram is trying to show how nutrients circulate in aquatic environments. Like we can see, the dissolved organic matter, particulate organic matter are all in the aquatic environment. So let's assume that this diagram represents an aquatic environment. We can see the phytoplanktons. So for example, this is seen in cococos. They are plant-like organisms which can be fed upon by bacteria. They are dissolved organic matter that can be taken up by bacteria. The bacteria can be taken up by the protozoa as particulate organic matter, and the protozoans can also be taken up by the zooplankton like shrimps and worms, whereas the fish, which are the top consumer in this aquatic environment, will feed on the zooplankton. Of course, nitrogen is a factor for phytoplankton to grow, but in a situation where you have excess nutrients, maybe there was rain and there was flooding and somebody has just applied fertilizer on the farm, and then the rain comes and flood everything into the rivers, what will happen? The nutrient is excess. So you begin to have excess growth of phytoplankton, then excess growth of algae, and then the algae might cover the surface of the water. And then bacteria and fishes will not be able to navigate to pick their own food in this food chain. So they may die or they may migrate to another environment. So water provides an environment for a wide variety of microorganisms to survive and function, but due to increasing industrialization and then our population growth, water have been adulterated with industrial waste, animal and human waste, leading to waterborne diseases. You will agree with me, even in this Lagos, look at the lagoon very close to us. It has become a kind of dumping ground or repository for different kind of waste. Even when this, what do you call them now, this waste, people that pack soak away, when they pack, all they do is to go back to the lagoon and dump it. all the malams that pick waste from your homes, where do you think they are taking it to? They dump it in the lagoon, or they dump it in the gutter as soon as it starts raining. Everything washed down to the lagoon and then finally into the ocean. That is why we started to have waterborne diseases. Okay, so what are the different kind of waterborne diseases that we could have that could be caused by microorganisms? Look at this table. You can have, for example, under the bacteria, you can have Salmonella typhi that causes typhoid fever. You can have Salmonella enteritis that can cause gastroenteritis. Gastroenteritis, the first symptom is the person will be gassing or belching with some feverishness and stomach ache. You can also have Helicobacter pylori. It used to be called Campylobacter pylori. These organisms give symptoms of ulcer, but then it can, if it persists too long in the intestine, it can lead to stomach cancer. Outside, in this country, when people present the symptom of ulcer, they give things like mismag to suppress it. But the truth of the matter is that is not the first thing that is supposed to do when you have ulcer symptoms, because it may not really be ulcer. It might just be the first thing to do really outside this climb. What they do is to do a test. There's breath tests, there are different kinds of tests that they can do to detect whether there is the presence of Helicobacter pylori in the stomach of that person. And so if there's Helicobacter pylori, the organism is treated with antibiotic first before then they can give other medications to suppress the burning sensation and gassing. So that if you keep giving mismag and the family, what can happen is that the organisms will be growing more because the organism also prefer that environment. And if it persists too long in the stomach, it can also actually cause stomach cancer. We also have Shigella dysentery that can cause shigellosis, Vibrio cholera that can cause cholera. Then Escherichia coli, that is the enterogenic strain, can also cause gastritis. And then we have the Legionella pneumophila that can cause pneumonia. But the viruses in water, you can take with water and that can cause diseases. You have the hepatitis A virus, which could cause hepatitis disease, and it's a terrible disease of the liver. Then you can have poliovirus that could cause poliomyelitis, that wasting disease of the bone. Then the Coxsackie virus disease that can also cause gastroenteritis. This disease in about 6 or 7 or 8 years ago really affected children, I think in the US or in the UK, I can't remember. I think some of the milk that were sold then, baby milk, got infected with this disease and it affected a lot of children. Then we have the protozoans. For example, the Giardia intestinalis, they cause giardiasis. Then the Entamoeba histolytica that can cause amoeboic dysentery. You know, dysentery can be caused by either amoeba, amoeba or some strains of bacteria. You also have the Naegleria fowleri that can cause meningoencephalitis in human. Then the algae, for example, you have the Goniolax catanella that can cause muscular paralysis, and then the toxicos that can cause Ciguatera fish poisoning. So I explained about algal bloom, which is caused by excessive nutrients in water. And of course that when there are excessive nutrients on this, in the water, it will lead to algal bloom and the growth of other water plants. And then it will cover the surface of water. It will prevent the sunlight from penetrating into the water. Therefore, the photoautotrophs that are supposed to be inside the water that should receive oxygen will not be able to do so. So most of them will either die or they migrate to a better environment. Then lack of penetration of oxygen will gradually lead to anaerobiosis in a situation where you begin to have anaerobic zones in the water body. And when you have anaerobic zones in the water body, it means that most of the aerobic organisms will either die or they migrate into other environments. So temperature can also change the water body. I told you to read about this, but I have explained most of it. What about hair microorganisms? Hair microorganisms. How do microorganisms get into our hair environment? Let's assume that you are sweeping and you did not wet the floor. As you are sweeping, some dust will carry some organisms that are present in that soil and release them into the environment. Let's assume the way some of you behave, you clean the chair and the table you want to sit. Then the same handkerchief, you dust it again, and then you dust it into the hair, and people that are sitting around you will inhale some of it. Look at those people that carry sewage all around. At times you see the excreta falling on the road. You see waste management and lawnmower vehicle. They carry dirt, and half of the waste they're carrying are already falling on the road, and some of these things are released into the environment. So those are the various ways. People sneeze. You sneeze without covering your sneeze, you cough without covering your cough, and these organisms are thereby released into the hair. When you go to the toilet, what happens? You want to flush. The ideal thing is for you to close the WC before flushing. If you do not do that, some of the organisms that are present in the excreta will follow the water aerosol into the hair, and some of them can actually survive in the hair for a long time, or people inhale it. Why is it that we are told to cover our nose? So that during this COVID, we will not release the organisms into the hair. And we are told to, use sanitizer. You open the door, you use sanitizer. You pick things on the table, on the floor, you use sanitizer, because these organisms, when they are when they are released into the hair, some of them have the opportunity to be able to stay long in the hair because they could survive the condition. While some will over time settle down on surfaces like table surfaces, the surface of the car, the surface of the chair, and so on and so forth. So what are some of these diseases that could be inhaled from the hair and could cause infectious diseases? For example, tuberculosis, that is caused by Mycobacterium tuberculosis, or the leprosy that could be caused by Mycobacterium leprae. Whooping cough is caused by Bordetella pertussis, and diphtheria is caused by Corynebacterium diphtheriae. Those are terrible diseases, but we thank God that at least for like Bordetella pertussis, the first 3 months when you give birth to a baby, the vaccine is already available. I think it will be DPT 1, 2, 3 or something like that. It's been a long time I did that. That is for diphtheria. So for almost of these diseases, there are already vaccines that has helped to suppress it. And that is why I keep wondering why people don't want to take COVID vaccine, because vaccines for things like smallpox, like Bordetella and diphtheria are even more difficult. You find it, they find it easy to take babies to punch their bum to take vaccines, but adults are running away from vaccines. It's so funny. So for bacterial tuberculosis, for example, that organism is dangerous because it eats up the lungs. It usually affects the lungs, and it is very difficult to treat. These are bacterium that has external capsule. And this capsule tends to protect them. So even when you take antibiotic, it does not easily kill the organism, because any organism that will take care of Mycobacterium tuberculosis, for example, must first break the capsule before they can penetrate the organism. And that is why you see people with tuberculosis, they cough for a very long time. And it may take up to 2 years or more for them to be totally healed. And that is if they adhere to the regimen of the prescription. And if it is very terrible, they are always placed in isolation ward, because this is a kind of disease that are easily transmittable. That is why you have to be very, when you go to the public restaurants, you look at the kind of restaurant where you want to eat using spoon, using cups from different people. You don't know who has tuberculosis, who does not have tuberculosis. And people with tuberculosis, they cough. When it is just starting, the cough might just be once in a while, or when it is going. But after a while, when it is getting to a very terrible stage, then you begin to see terrible cough and sputum with temperature. And that is why some of them don't survive. But right now, the drugs are working. And if the person follows the regimen, the person is most likely to survive. Can you imagine also the leprosy? You see people with leprosy, you see how that Mycobacterium leprae has eaten their hand. If you see some Hausa people without fingers, it is the work of leprosy. It attacks the eyes of some people, and you will see the eyeballs terribly eating up. You see the flesh. So these are terrible microorganisms, and they are very, very contagious. So you also have some organisms that you can contact from the head that can cause first, like skin allergies. And when the allergy starts and itching starts and you begin to have sore, if the sore refuses to heal, if the person has diabetes, for example, and the scratching leads to sore and the sore is failing and refusing to heal, it will resolve to cancer of the skin at the end of the day. So we have microorganisms outdoor, not only outdoor, we also have indoor. So as you have microorganisms outside, you also have microorganisms inside. So the outdoor microflora depend on the season, the temperature, and the environment. What do I mean by that? If you are familiar with K2 area, maybe Ojota, K2, and you're on your way to the Express 7-Up area, you'll be familiar with the very big dump sites there. So that outdoor, people living around that area, for example, you cannot compare their outdoor microflora with people living in the GRA. Definitely, if you perform a test, the microorganisms you will find in the outdoor area of Ojota or K2 will be more terrible than the one you will find around GRA or Ikoyi. Actually, we've carried out that test before with some of my MSc students about 4 years ago. So some of them took sample near the dump site at Ojota. Some took sample at Oshodi Market that was so crowded. Then some took at GRA Ikeja, some took at Ikoyi. And like we imagined, both the fungi and the bacteria are terribly dense at Ojota, then followed by Oshodi Market. So the season also matter. The temperature matter. When the temperature suits the microorganism, they grow more. During the heat period, you discover that we sweat a lot, and then we tend to have itching of the body more. But during the rainy season, when the rain starts, the rain falls on the soil, and then some organisms, as the rain hits the soil, some organisms are released into the air. And that is why at the onset of rainy season, people tend to have cold and cough. Okay? So you can have positive bacteria You can have negative bacteria, especially bacteria that can resist drying. Those bacteria that can survive with little amounts of water can actually survive in outdoor. Now we also have some indoor microflora. For example, you have the Aspergillus fumigatus that can cause asthma in human beings and also cause mycotic abortion in cattle. So you have different fungi indoor, especially if you live in an environment that is humid. Like some of us whose office is resident at the, what do you call it, diploma building, right inside the lagoon there. The lagoon is a salty environment. Sometimes we leave our table and chair for 5 days, and when you come back, you see fungal growth all over your table and your chair. Then you take some spirit, you clean it up. By the time you come back again after the weekend, they're already growing. That is the environment. But that does not happen if I find myself in an environment that is not too close to the lagoon. So how do you detect microorganisms in the hair? The simplest and the common or conventional way of detecting hair microorganism is to expose your agar. For example, if you want to detect fungi in the hair, then you prepare PDA, potato dextrate agar. That is the general agar for detecting fungi. You will soon find out by the time you start your practical. You expose it, you look for a table or a surface that is higher than your head, maybe on the roof, because you want to study hair microorganisms, so you don't want any other activity to interfere. So you can put it on the roof or on any surface that is very tall. Then you prepare your agar, you expose it for like 2 to 3 minutes, you close it back, wrap it and take it to the lab and grow. Then you study the organisms that is able to grow on the agar. If it is bacteria you want to detect, then you have to prepare plates of nutrient agar. And you put them at different sites, not only in one place. You have to take them, give some gap and take maybe 4 or 5 plates and then take them. But we also have some equipment or samplers that are used to count airborne microorganisms. You have spore load in the hair that are detected by Anderson sampler, rotor sampler, and Burkhardt spore sampler. Now, these days, we have different kind of sampler that will sample the hair. It will draw the hair. There's a button you will press, and then it will draw the hair, whether indoor or outdoor. It will draw the hair and then collect the microorganism. And it is like an incubator. It will also incubate, and some are even automatic. That after incubation, it will even tell you what the organism that you have in there are. What are common examples of aeroallergens? Aeroallergens could include microorganisms, it could include spores, it could include pollen. Anything that causes allergic reaction is aeroallergens. So for the microbes, we have fungi. And some of the fungi that can cause allergic reaction, some of the common hair organisms include Aspergillus, Penicillium, Fusarium, Candida, and Alternaria. Some Aspergillus and Fusarium, not all of the species though, but some of them actually have dangerous toxins that if you keep inhaling them, after a while, then the person might develop some kind of cancer when there's excessive dose of that toxin in the body. It may attack the lungs or the liver. Then the algae, we have the Chlamydomonas, the Nostoc, the Trichia, and the Oscillataria. So the lichen, we also have the Cladonia and Heteroderma lichen. You already know that lichen is a combination or relationship of 2 organisms from your secondary school biology, fungi and algae coming together to form lichen. Some people call it leaching. So what are other undesirable roles of microbes in the environment? We have talked about diseases. Samuel Oladele, can you please unmute and tell me what's your question? Oladele Samuel, what's your question? All right, I'll just continue. Maybe later you will talk. Other undesirable roles of microbes in the environment, so apart from diseases. Corrosion of pipeline is caused by species of disulfovibrio. So you have oil pipes and different pipes that could be corroded, especially in those days where we use iron or steel pipes. You also have contamination of drilling fluids by bacteria. For those that are working in the oil company, essentially, corrosion of oil and contamination of drilling fluids are majorly caused by the activity of bacteria. Then contamination of painted walls. You see some walls, you see green color, yellow color, black color. These are different species of fungi that can grow on painted walls because the paint there contain the nutrients. You know, the constituents of the paint are a kind of nutrient that support their growth. And definitely that environment favors them because fungal species, they like acidic environment and they do not need much water to grow. So if the place is humid and there's temperature, then fungal species will grow appropriately. What are the desirable roles of microorganisms in the environment? So microorganisms, like I said earlier, is not all about bad, or diseases. You could also use them, just like we discussed under food. So increase in environmental contamination leads to progressive deterioration of environmental quality. But however, microbes play some roles in the preservation of the environment. We talk about oil spillage, we talk about metal contamination, contamination with chlorohydrocarbons. What can we do about it? That was how the issue of bioremediation comes in. And when you talk about bioremediation, that is the use of living thing to kind of clean the environment. So that suggests to you that it is not only microorganisms that can be used for bioremediation. We also have phytoremediation under bioremediation, and phytoremediation has to do with plants, using plants or plant derivatives to remediate the environment. But we are microbiology here, we are talking about microbiology. That we have botanists that will talk about phytoremediation in their own course. So for bioremediation, you know, we use normal or selected or genetically engineered microbes or their products like their enzyme to clean up the environment. So, and we can do that through various means like processing biological waste and avoiding microbial pathogenesis. When we say we produce biogas from waste, that is one of it. We simply pack the waste and then apply some microorganism and convert the waste into various value-added products instead of the waste to remain in the environment, causing nuisance and diseases. So you actually use the waste with the activity of some microorganisms and you convert them to biogas. I'm working on biogas. I'm also working on bioconversion of waste to bioethanol. And I've also worked on the bioconversion of waste to biochemicals. So those are the useful things you can do with microorganisms. Then concentration of metals and radioactive materials to avoid toxicity or to recover metals for reuse. That is bioaccumulation. In a situation where you have radioactive compounds released into the environment, and metals, you know, it's not all metals that are good. So you can actually deploy microorganisms into that environment we have microorganisms that usually use metals as their own substrates, that is, as their own food for growth and to carry out their metabolic activities. So these microorganisms can take up these metals for growth. So if you need your metals back, you can also take these microorganisms and then break them and then collect your metals back. You allow the metals to be accumulated in the body of microorganisms. They use the metal as food. They now grow bigger. Then you don't need to collect, if you need to collect your metal back, you recover it from the microorganisms. We also have biodegradation of organic chemicals. For example, polychlorobiphenyls, PCBs, they are not good in the environment. So microorganisms can be used to remove them from the environment. Then the waste site cleanup operations, microorganisms are now sprinkled on waste sites, depending on the kind of waste, and then they clean the environment. Like I talked about the biogas using anaerobic digester. Anaerobic digesters contain microorganisms adapted to grow and multiply in the absence of oxygen, and they can survive the environment. So they can digest waste and then give you methane in return, which can be used for cooking or for heating. And nowadays it is being converted into electricity. So nutrients are converted to microbial biomass, methane and carbon dioxide are consumed, are released. The liberated methane is used to heat the digester. The sludge released by the digester has reduced malodor. So at the end of anaerobic digestion for the production of biogas, for example, the remnant does not smell. So if you pack all the cattle dung, the waste from the poultry, and you convert them to biogas, the that is remaining, they will not smell. And then you can also use them as organic fertilizer. We actually use some of these residues as organic fertilizer, and they really performed well on some vegetables. We also have the use of microorganisms to clean marine oil spills, jet fuels, and organohalogens in groundwater aquifer, especially in the US. They have really used microorganisms to clean groundwater. For some of you that will carry microorganisms later to 400 level, we also have a course that is called petroleum microbiology. Under this, most of these things are studied. So microorganisms are used in oil prospecting and recovery, and also in biogeochemical cycling. For example, microorganisms are involved with carbon cycle, sulfur cycle, and phosphorus cycle. If those of you that will do microbiology in 300 level, you will study this the more. This is just basic kind of introductory microbiology. We also have degradation of lignocellulose, that is an agricultural waste for the production of feeds and other value-added products. Like I told you earlier, I've used some of my microorganisms, which I isolated since 2008. 2008, I'm still using them to convert some waste to produce value-added products like enzymes and like biochemicals that can be used in other industries. So how do you control microorganisms in the air? You suppress dust. For example, as common as you want to sweep your environment, maybe your compound, and it is not cemented. The first thing you are supposed to, even if it is cemented, during dry season, there are dust. So what you are supposed to do is to first wet, you suppress the dust by wetting. Our forefathers do not know that there are microorganisms in here, but you will discover that when they want to sweep in the village, the first thing they do is to use water to wet the ground. Probably they have over time studied that whenever they sweep, they begin to cough and so they have developed a means of wetting the floor first before they sweep. So you can suppress dust even with common wetting of the floor before you sweep. But there are, technological way of doing that these days. You can also inactivate microorganisms using chemical agents. Then you can inactivate microorganisms by using ultraviolet rays. That is, if it is within a room. For example, the theater And we also have our own room where we do carry out our inoculation. Most of the time we put on the UV lights to work for a while, then you off it back before you begin to work. You will be sure that all the indoor microorganisms have been killed before you carry out your work, especially those that are working on DNA and RNA studies. Then by the use of filters, for example, now even we are using nose masks to prevent inhalation of microorganisms. Okay, we also have soil microorganisms. Soil microorganisms abound. So I just mentioned a few. For example, we have those that are common to the soils are the ones I mentioned. That does not mean you will not find other microorganisms in the soil. Any microorganisms can be in the soil. It's just that a kind of soil might have one kind of microorganisms than the other. For example, if you have acidic soil, it is not all type of microorganisms that can survive in acidic soil, but you have microorganisms that are acid loving. So they can survive that environment. They are called acidic microorganisms. You have, I mentioned that to you even in our last lecture. So the common bacteria in soil include actinomycetes, and you have a lot of different actinomycetes, but the common ones include streptomyces. You have the nocardioform type of actinomycete called the nocardia. You also have mycobacteria in the soil. For example, mycobacterium, which could cause tuberculosis or which could cause leprosy. But you have other type of mycobacterium that are non-pathogenic. It's not all mycobacteria that are pathogenic. You also have the corynephrums, the Corynebacterium, Atrobacter, Cellulomonas. Some of them are useful. For example, the Corynebacterium can be used to produce some substances, and the Cellulomonas, most of them can produce enzymes that are able to break down cellulose for the production of value-added products from plant waste. Some common fungi in the soil includes deuteromycetes, zygomyces, ascomyces, and whoromyces. I'm sure you will study that in botany. Then the common algae include Chlamydomonas, the chlorophyta, and the xylophyta. Most of the soil microorganisms get into the hair and they can be hail, causing hairborne disease, as I explained earlier. Some can be washed into the water bodies by flooding during rainy season, and when consumed, could lead to waterborne diseases. So what are the beneficial roles of soil microorganisms? The reduction of atmospheric gaseous nitrogen to ammonia is called nitrogen fixation. So in the soil, microorganisms carry out nitrogen fixation. And this can occur in whether free-living bacteria, for example, the Clostridium, the Klebsiella, and Methanococcus. It can also take place in bacteria living in symbiotic association with plants, such as legume, for example, Rhizobium. We all know about Rhizobium right from our secondary school days. In cyanobacteria, that is Nostoc and Anabaena, they play important role. Soil microbes are involved in nutrient cycling, like I said earlier. Then you have the mycorrhizas, in a situation where you have relationship between plant roots and fungi to form mycorrhizae. Plants, sometimes when there's deficient water and nutrients in particular soil, they form relationship with fungi because fungi also have root-like algae, which can burrow deep into the ground to draw water and then pass it on to the root of the plant. And then the root of the plant pass it to the body of the plant. So that is the work of mycorrhiza. It's a further work in 300 level. You also have what you call the rhizosphere in the soil. This is the region surrounding the plant root where the nutrients such as various alcohol, ethylene, sugars, amino acid, organic acid, vitamins, nucleotides, polysaccharides, and enzymes are released from the roots of the plant. And this increases microbial population. What that is trying to say is that the regions surrounding the plant root is called rhizosphere. And this rhizosphere has been studied extensively. And in the rhizosphere, we have microorganisms. That is not far-fetched because the region of the rhizosphere has different substances and nutrients, like I have listed: organic acid, vitamins, polysaccharides. So these organisms can pick any substrate they like around that region. So microorganisms like to survive, like to play, like to live around the rhizosphere. We also have the rhizoplane, the plant root surface, which also provide a unique environment for some microorganisms. We have some microorganisms that are plant growth promoting microorganisms. For example, the rhizobacteria, which include the genera Pseudomonas and Acromobacter. What are the environmental influences on soil microflora? That is, what are the factors that can influence the presence of microorganisms in a specific environment? Moisture content, I've said that. If you have high moisture and water available, you are more likely to see more bacteria because they require more water to survive, like we discussed under food, compared to fungi. But if you have sufficient moisture, like high humidity, and the temperature is hot, then you will find fungi growing on your food, on your wall, on your belts, on your shoes, on your leather bags. Then you also have neutral alkaline environments. Then the temperature, like I told you, temperature has a lot to do. Temperature and moisture has a lot to do with the kind of microorganisms you will find in the environment. Of course, the oxygen content, because the organisms that like air will survive where you have enough oxygen. And those that do not like oxygen will survive where there's no oxygen. But you also have some microorganisms that are called facultative aerobes. They are facultative aerobe, that is, they will prefer anaerobic environment. But if there are air, no, they can still tolerate it to some extent. The same thing, you have facultative anaerobes. So you can read about how root nodule is formed in rhizobium, though that is not part of your scheme, but it's just interesting. Okay, what about in the industries? Paint industries, shoes industry, whatever kind of industry that you want to talk about. In the industry, of course, we have positive roles, that is desirable roles that microorganisms can play there. We also have undesirable roles that microorganisms can play in the industries. So let's look at some of those desirable roles, which we have actually also mentioned under food or under environment. That is because industry cross across. If you are talking about food industry, you are talking about pharmaceutical industry, you are talking about the environment, no, you are talking about oil and gas industry. So you have various types of industries. Whichever industry you want to talk about, whether it's pharmaceutical, food, or environment, microorganisms are there. Microorganisms cut across, that is, microbiologists cut across any field of endeavor. So what are the desirable roles? Production of biofuel, like I said, ethanol from agricultural waste. I have done that. I have actually produced bioethanol using some Streptomyces species, and the paper is published, it's online. I've produced biogas, no. Then production of biodiesel, that is renewable fuel made from pure or used vegetable oil. So if you have some mechanic oil, some vegetable oil that are dirty, that are supposed to be poured into the drainage that would affect microorganisms, this can be converted to biodiesel. Biogas, like methane, like I told you, produced by anaerobic organisms. You also have bacterial biofertilizers. Apart from using the remnants of biogas to produce biofertilizer, You can also use live organisms, to produce biofertilizers. For example, the rhizobium can be used, to coat seeds, to treat seeds, and then the seeds will produce the kind of character you want in the plant. They have one at IITA Ibadan. They have one whereby rhizobium, as bacteria is being processed in large quantity, and they are used to coat beans or groundnut and sold to farmers. So they make at least it increases the farm yields by about 40%. And they also have some other mini industry there where it is called Aflasane. Aflasane, they produce aspergillus species that usually attack corn in the store. So they isolated these aspergillus species, bioengineered them, and coat them on millets, on groundnuts, and maize, and so on and so forth. So that by the time that maize is grown, then it is protected from being attacked by aspergillus species. So you always have disease-free yam, disease-free corn, disease-free groundnut, and so on and so forth. We have phosphate solubilizing microorganisms. We know that phosphates are always in the soil, but in the form they are in the soil, they cannot be taken up by plants. So it is the work of some of these microorganisms that are able to solubilize the phosphate that will help to solubilize the surface that are locking in the soil so that plants will be able to take them up. So some of the microorganisms that can solubilize phosphate include Pseudomonas, Bacillus, Penicillium, and Aspergillus that can convert insoluble phosphate to soluble and usable phosphates. We have production of antibiotics. For example, streptomycin can be produced by Streptomyces griseus. And these days, people are beginning to run away from synthetic antibiotics because of antibiotics. So it is coming up again, the use of microorganisms. Microorganisms were the first producers of antibiotics before pharmacists begin to produce synthetic forms. So people are beginning to go back to antibiotics again. The manufacture of commercial valuable chemicals such as acetone, lactic acid, dextrins, glutamic acid, used in food and pharmaceutical company. Around 2019, I visited Germany and I used some of our ways to produce chemicals. I produced lactic acid, I produced gluconic acid, and some other acids that I don't want to mention here. Microbial enzyme production, for example, lacases, which are used in the production of a variety of products, whether in the food industry, in the cosmetic industry, or where they produce detergents. Most of these enzymes are useful, not only lactase, amylase, they are useful in the food industries. And these enzymes can be released by microorganisms. When you get to 400 level, for those of you that will go for that, you will learn how to produce enzymes. Used in baking industries. They are used in baking industry. We all know that yeast are used to improve dough for production of bread, cakes, and other pastries. The microorganisms can be used as bioinsecticides and biopesticides. For example, there's a popular bacteria, Bacillus thuringiensis, that has been tried and used successfully for the production of insecticides. What are the undesirable roles of microorganisms in the industries? I've mentioned this earlier. They cause spoilage or biodeterioration of industrial products, for example, leather. Organisms such as Micrococcus luteus, Penicillium, Aspergillus, and Deuteromyces can eat up leather. That is why if rain beats you, for example, and you were actually carrying leather bag, wearing leather shoe or belt, and then you now get to your room and you put those things inside your wardrobe and lock your wardrobe, and then you travel home for weekend. By the time you come back 3 or 5 days later, you will discover some whitish growth on your belt or your bag or shoes. Those are organisms. And if you leave it for too long, they will begin to eat up the leather. So they also eat up some textiles, some clothes. For example, Bacillus megatherium, Cellulomonas, Corynebacterium, and Alternaria, they can spoil textiles because they can eat them up. The jute plants, that are used to produce bags, they can be spoiled by Atrobacter, Bacillus, Xanthomonas, and Cytophaga. Painted surfaces can be spoiled by Aspergillus, Flavobacterium, Plasmodium, and Pseudomonas. I hope you can still hear me. If you can hear me, please say yes.
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 All right, thank you very much. Because they just
Yes.
then rubber, for example, rubber can be spoiled by Pseudomonas, Nocardia, and Penicillium. Of course, pulp and paper. Some of us that graduated since 1993 or 1994, if we pick up some of our notes, you just see that some of the notes inside are already punctured. Some are having pinkish or reddish color or blackish color. Microorganisms have actually invaded them. The microorganisms that can eat up paper or destroy paper is not only Chaetomium. We have different kinds of bacteria and fungi that can actually destroy papers. They also cause food deterioration in the industry. For example, those that bake bread molds often disturb them. For example, Rhizopus stolonifer can cause ropiness in bread and cake and other bakery products. Okay. All right, so I think we want to stop here. We want to stop here for today. And for this next topic you are seeing is no more for me this semester. I taught it last year. So we are very fine because this is 12:15 stop for today. And then we'll move into the next topic on Tuesday. So but that is where the challenge is. I always have problem with 8 o'clock lectures for students. I imagine those that are living outside the campus, how they will face the traffic to get here, even those in the hostel, how you queue up in the bathroom and all that. But the truth of the matter is we just have to meet on Monday morning, 8 to 9. So you will try your very best. And you are lucky that this time around it is online. So nothing should stop you. Nothing should stop you from actually attending your classes on Tuesday morning, 8 to 9. So I will pass the message to your class captain if the practical will start on Monday. Because on Tuesday, sorry. Every Tuesday, there's supposed to be 1 hour lecture, then practical 9 to 12 or thereabout. And they are going to share the classes because they have not yet migrated you people into the LMS. I've not checked since yesterday, but I've written officially that they should migrate you and delete the last year student. As soon as they do that, I'll be able to know how many students are registered for MIC 211, and then I will communicate that to the lab technician. So they are ready. It's just for them to share you into group, and then you know which group will start first. But if you have an idea of the number of students, for example, Evans, the class captain, can tell me these are the number of people for MIC 211. It's a compulsory course for all microbiology students. So you can tell me how many. If Evans, you can also get in touch with the class captain for botany, class captain for cell biology, for zoology, and then biology education. You can tell me how many students, about how many you have in each of your class. Then I'll calculate together and pass the message to the lab attendant. I know it may not be accurate, but it will give them an idea of how many you are, so they will know how many groups they are going to share you into. Am I clear? So we'll meet on Tuesday morning, 8 o'clock, by the grace of God. God will keep you safe and strong. Thank you very much. God bless you. Bye-bye.
Thank you, ma'am.
All right, ma'am. Thank you, ma'am.
Thank you, ma'am.
Thank you, Ma.
Thank you so much. God bless you.
We appreciate your lecture.
Yeah, thank you so much. I appreciate you all. Thank you. Bye-bye. God bless you.
Bye, ma'am.